Hi, my name is Greg. I'm an MD PhD candidate from the Neuropsychiatry Lab in Hadassah and Kalem. And I'll be presenting our work on disorientation in the context of Alzheimer's disease. No. Oh, okay, there we go. So what do we talk about when we talk about Alzheimer's disease? So Alzheimer's is essentially being recognized as a global health issue, as it's estimated to affect 115 million people in 2050. And despite decades of research, the relationship between the primary histopathological findings remain a matter for debate. On the other hand, neuroimaging is becoming more and more essential in both clinical practice and research of Alzheimer's disease. And finally, we can say that the core disturbance in Alzheimer's is still a matter of, kind of remains ill-defined. And which brings me to my last point, the last one brings me to this slide. And in our lab, we consider orientation to be kind of very highly connected and related to Alzheimer's. And essentially, we have a more kind of elaborate, a more inclusive definition of orientation than commonly used in the clinical practice, and it's based on a paper by G. Barrios from '81. And it's essentially, we consider this orientation to be the tuning between the subject and his environment to his mental representations of the same environment. And this kind of concept can be very easily applicable to space, to the domain of space, kind of evoking this egocentric, allocentric reciprocality, right? The places we see and the mental map we form of the said places. But this could also be applied to more abstract domains like the domain of time or the social domain. And just before going any further, I would like to stay out our research questions. So we're asking, how does the increasing pathological burden across the Alzheimer's disease spectrum affect the pattern of brain activity and orientation performance? And we've generated essentially two hypotheses to answer this question. So the first hypothesis kind of described this monotonically decreasing process where an increase in pathology would result in a decrease in both behavioral performance and in brain activity. An alternative hypothesis is essentially a two-phase model where an initial accumulation of pathology would bring out a reactive phase resulting in hyperactivation or neural hyperactivity. And once that compensatory mechanism is exhausted, we'll get the declining slope and even possibly a more rapidly declining slope that will result in cognitive, essentially cognitive disturbances. To test orientation in the context of these questions, we use the task we developed in our lab, and the task is very simple. Essentially, we present subjects with pairs of either places, events, or people, and ask them to indicate which of the two is closer. So, geographically closer if these are two cities, chronologically closer if these are two events, or personally closer if these are two people. And other than the task, we essentially established a very kind of ambitious pipeline together with our colleagues at the PETMR unit at Assuta Medical Center that includes both functional and structural imaging. And we applied this pipeline to three relevant clinical groups. So we have the Alzheimer's disease patients, we have the age-matched healthy controls, and in addition we have a third group, which is the MCIs, the mild cognitively impaired patients. And these are patients that are not demented, but they exhibit a cognitive decline. This could be considered a prodrome, right? A prodrome of Alzheimer's disease. And these three separate groups underwent this pipeline, and I'll show some of our results. So first of all, let's look at the behavioral results. So we look at the behavioral performances of the orientation task that I've just presented. And we're looking at these success rates. And if we look at the task, we see that essentially the task is successful in separating the three clinical populations. But looking at our trials in a higher resolution and segmenting them into proximal and distal trials, meaning asking people about the places, events, people that are close to them, as opposed to the places, people, and events that are further from them, we see that what's driving this separation is the more distal subset. We can consider that to be the more difficult subset. So that's one way of looking at it, but alternatively, we can think about, especially this group of NCIs, that for the proximal subset was able to compensate and perform on control level and essentially produce control level performances. So this is very interesting. They're able to somehow to compensate for their kind of initial brain pathology. And just anchoring our work in kind of classic results, the black silhouette is essentially marking the brain regions where Alzheimer's disease, as opposed to healthy controls, would have less probability of having gray matter. This is a VBM analysis, and this is an indicator of brain atrophy. And these are very classical Alzheimer's disease related regions. We have medial temporal regions and parietal regions and even frontal regions. So that's the silhouette. The bar graphs represent essentially the analysis of the PET-FDG, which essentially is a measure of consumption of glucose, right? And we see that, and the logic being that the less a brain region is consuming, the less it is active, metabolically active. And we see again a decreasing trend, although not always significantly different between healthy controls and NCIs, but we do see a trend going from controls to Alzheimer's disease patients. And considering these two in the context of our previously mentioned hypothesis, we see that these two kind of match our first hypothesis, referring to a monotonically decreasing process. Now let's look at the functional fMRI results that essentially we got while subjects were performing the task. So just before going into the results, I'll state that this was an ROI-based analysis, meaning that we looked at predefined regions. And what we did was took another control sample and defined the task-evoked regions using that control sample to avoid cyclicality and essentially, finding exactly what we're looking for. So we took another control group and we defined these orientation evoked regions. And these could be, and we did work on that, they could be segmented according to the three domains, the space, time, and person domains, presented here by the different colors, and here using a different color scheme. Sorry for that. No, we don't see my mouse. Here using a different color scheme. But what we look at in the current context, we'll be looking at this general region of orientation, which includes these regions marked by yellow that essentially are aligned on this parietal hippocampal axis that includes our splenial cortex, precuneus, parahippocampal, and hippocampal regions. So this is kind of a domain non-specific regions or domain general regions we looked at. And that's how we define them. And just looking at the results, and these are the data, we see a very interesting pattern that's different from the pattern we've just seen. We see some base level for healthy controls, and then upregulation of activity during orientation performance in these orientation evoked regions, followed by a decrease for Alzheimer's disease patients. And this kind of matches our second hypothesis, the two-phase hypothesis. To try to understand what's driving this activity, our first hypothesis was that it has to do with compensation. Essentially, subjects are trying to compensate for their brain pathology and essentially upregulating their activity. And to kind of probe at that point, we again segmented our trials according to the levels of difficulty. So here we segmented them into 4 levels of difficulty. And what we see for both healthy controls in green and Alzheimer's disease in yellow, we see no difference or no correlation between essentially the level of brain activity and the task difficulty. And for healthy controls, for us at least, it implies some sort of a ceiling effect, right? The task is equally easy no matter the distance. And a floor effect for Alzheimer's disease patients, meaning that the task is equally hard no matter how close the stimuli are. But for the MCI marked in blue on the top, we see an interesting correlation going from the first level of difficulty to the third level of difficulty, and then a break in that correlation. Incidentally, that break between the third and fourth levels of difficulty match the reduction in performance, or I've showed at the behavioral results. So kind of combining these two results together, what I'd say would be that the MCI subjects are able to compensate and upregulate their activity up to a point, but once they reach some sort of threshold, their activity drops, and that's evident in their behavioral performances. And this is the break between 3 and 4 is exactly the distal and proximal stimuli I mentioned earlier. So these results kind of led me to believe that this is indeed some sort of compensatory mechanism that essentially MCI patients are kind of employing to remain close or at healthy level controls. And this could be possibly contributing to their ability to kind of stay below the threshold of detection and essentially kind of get diagnosed later in life. So just to kind of wrap up, considering the healthy control to MCI to Alzheimer's disease trajectory, we've seen subjects exhibit kind of the expected metabolic decline. However, in considering fMRI orientation-evoked activity, we see an upregulation for MCI and then a decline for Alzheimer's disease. And there are several possible explanations. One could be a dysregulation in the system, right? The system brain regions are known to be regulating each other, so some sort of damage could affect regulation and would result in hyperactivation. An alternative explanation would be some sort of compensation. And this last point for me is extremely important, and it's essentially what's driving this project forward. There are some evidence that hyperactivity in the context of Alzheimer's disease is not an innocent bystander, but itself plays a role in the pathological cascade. So essentially, reducing that hyperactivation could have some sort of therapeutic implications. And that's, I think, this is kind of the question we're grappling with, and the question we're using our multidimensional data to try and answer. And that's the question I'll be leaving you with. And I would just like to thank my lab members and our collaborators from the Assuta unit and the National Institute for Rehabilitation. And you, of course, for your attention. Thank you.